Hello and welcome back to Edu Pharmacy. Today, we're exploring a fascinating yet often underestimated subject in pharmaceutics: powder characterization in the real world. At first glance, powders may look simple, just tiny particles of solid material. But in reality, they are surprisingly complex to handle. Their behavior can be unpredictable, and even small changes in particle properties can make a huge difference in how a medicine performs. Think about this. More than 90% of drugs are handled in powder form at some stage, whether as raw ingredients, intermediates, or finished products like tablets, capsules, and inhalers. If we don't fully understand how these powders behave, we risk producing medicines that are inconsistent, unstable, or even ineffective. That's why powder characterization is so important. It helps us ensure every single dose of medicine is safe, effective, and of the highest quality. Let's start with the basics: particle size and distribution. This is one of the most critical parameters. Smaller particles usually dissolve faster, which is useful when a drug needs to act quickly. But very fine powders can have poor flow and may clump together. On the other hand, larger particles often flow better but dissolve slowly, which could delay drug absorption. So, in practice, scientists don't just measure the average size. They look at the distribution of particle sizes in a sample. A wide distribution can lead to segregation during mixing, while a narrow distribution gives better uniformity. Common techniques used include sieving, laser diffraction, dynamic light scattering, and optical microscopy. Now, size isn't the whole story. Particle shape and morphology are equally important. Imagine filling a jar with marbles compared to filling it with needles. The marbles pack neatly and flow easily, while the needles interlock and leave gaps. The same principle applies to powders. Spherical particles tend to pack and flow better, while irregular or needle-like shapes can cause problems during mixing and compression. To study this, scientists use scanning electron microscopy, or SEM, which produces highly detailed images of particles and their surface features. This helps predict how the powder will behave during processing. Next, let's move to surface area and porosity. A larger surface area usually means faster dissolution, which is particularly important for drugs that are poorly soluble. That's why micronized or nano-sized powders are often used. They expose more surface to the solvent and improve bioavailability. Porosity also matters. A porous particle can absorb more liquid, which affects how it behaves during granulation or when compressed into tablets. To measure surface area, one of the most widely used techniques is the BET adsorption method. Another key property is flow. In large-scale manufacturing, powders need to flow smoothly into tablet presses and capsule filling machines. Poor flow can cause inconsistent fill weights, which leads to dosage errors. To evaluate flow, we look at parameters like the angle of repose, Carr's index, and Hausner ratio. Each of these provides insights into compressibility, cohesiveness, and flowability. If a powder has poor flow, formulators might use granulation or add flow enhancers such as magnesium stearate. Let's not forget about moisture content and hygroscopicity. Some powders easily absorb water from the environment. This can lead to clumping, caking, or even chemical degradation. For example, certain antibiotics and effervescent formulations lose their effectiveness if exposed to humidity. To measure this, techniques like loss on drying and Carl-Fisher titration are commonly used. Understanding how much moisture a powder can absorb helps in selecting the right packaging, whether it's blister packs, HDPE bottles, or containers with desiccants. So, where does all of this matter in the real world? Let's look at some applications. In tablet manufacturing, particle size and flow ensure that every tablet has the right dose and dissolves consistently. In inhalers, particles must be between 1 and 5 micrometers to reach the lungs effectively. Larger particles get trapped in the throat and smaller ones may be exhaled. For injectable suspensions, particle size and shape determine whether the drug stays stable or settles out. And when it comes to packaging and storage, powders that absorb moisture need protective containers to maintain stability. In short, powder characterization is not just about lab tests. It directly affects patient outcomes. Looking at the future, modern technologies are transforming this field. X-ray diffraction, or XRD, is used to detect different crystalline forms known as polymorphs, which can drastically change solubility. Atomic force microscopy gives us nanoscale surface details, and real-time particle analyzers provide continuous monitoring during manufacturing. These innovations are part of what's known as Process Analytical Technology, or PAT, and they help make pharmaceutical development faster, more efficient, and more predictable. To wrap it up, powder characterization is the backbone of pharmaceutical product development. Every property, size, shape, surface area, porosity, flow, and moisture plays a role in determining how a medicine performs. By carefully studying these parameters, scientists can design formulations that deliver consistent results, meet regulatory standards, and most importantly, keep patients safe. Thank you for joining me in this deep dive into powder characterization in the real world. If you enjoyed this video and found it helpful, don't forget to like, share, and subscribe to EduPharmacy for more pharma insights. See you in the next video.